Disaccharides. Disaccharides are compounds containing two monosaccharides. Di means two. Okay. These two monosaccharides are produced when these disaccharides on hydrolysis with dilute acids. On hydrolysis with dilute acids or enzymes. Okay, gives these two monosaccharides. Okay. Such compounds are called as disaccharides. Okay, and these two monosaccharides are joined together by an oxide linkage. By an oxide linkage, and this linkage is called as glycosidic linkage. Okay, glycosidic linkage. Different examples of disaccharides are sucrose, maltose, lactose, etc. Okay. Let us discuss one by one, of which sucrose is the most important one. Let us discuss the structure of sucrose. Okay. Sucrose. Sucrose is ordinary table sugar which we are using in our daily life. Okay. Sucrose is table sugar. It is present in sugarcane, sugar beet, then pineapple, banana, etc. Okay. Let us discuss the structure of sucrose. Structure. Its molecular formula by elemental analysis and molecular determination shows that its molecular formula is C12H22O11. Okay. This sucrose on hydrolysis with enzymes or with dilute acids. Enzyme used here is invertase. So, in the presence of invertase, this sucrose on hydrolysis, okay, gives D plus glucose and D minus fructose. Okay. So, equimolar mixture of D plus glucose and D minus fructose are obtained when sucrose on hydrolysis with invertase. Okay. See the reaction. C12H22O11 on hydrolysis. One water molecule is removed. Okay. During hydrolysis, one water molecule is removed. Disaccharides on hydrolysis, one water molecule is removed and two monosaccharides are produced. Here C6H12O6. This is D plus glucose. And C6H12O6, this is D minus fructose. Okay. Equimolar mixture of these two compounds are obtained. Okay. Next, different observations and reactions shows that the structure of sucrose contains a glycosidic linkage between C1 of alpha D glucose, okay, C1 of alpha D glucose and C2 of, that is second carbon, and this is first carbon of beta fructose. Okay, since the reducing groups, these reducing groups are involved in the linkage formation. Okay, These reducing groups like CHO and CO groups present in the glucose and fructose are involved in the glycosidic linkage. Okay, glycosidic linkage. So they are not free for reactions. So these sugars, this sucrose is also called as non-reducing sugar. Okay, it's also called as non-reducing sugar. Okay, this is the structure of sucrose, and sucrose is dextrorotatory. Okay, so this is the alpha D glucose structure and beta D fructose structure, and there is a glycosidic linkage between C1 carbon atom of alpha D glucose and C2 carbon atom of beta D glucose. Okay, this is pyrano structure of alpha D glucose, and this is the furano structure of beta D fructose. Okay, in the case of sucrose, it is dextrorotatory. Okay, on hydrolysis, it gives two monosaccharides, that is D-glucose. D-glucose is dextrorotatory. Okay, that we know, and gives beta D fructose. Beta D fructose is levorotatory. Okay. Okay, and in the case of levofructose, it is the optical rotation is minus 92.4 degree, and in the case of dextrorotatory D-glucose, the optical rotation is plus 52.5 degree, and here we can see that the value of D-fructose is more. So after hydrolysis, the compound is becoming a levorotatory compound. Okay, so after this hydrolysis, this dextrorotatory sucrose gets converted into levorotatory. That is, the sign of the compound changes. So this process, this inversion of sugar is taking place, and this inversion is known as inversion of sugar. Okay, inversion of sugar. Okay, inversion of sugar takes place, and the resulting mixture is known as invert sugar. Okay, resulting mixture is known as invert sugar. Okay, next compound is maltose. Second one, maltose, also called as malt sugar. Okay, it is seen in sprouted barley seeds. Okay, it does not occur free in nature, and it is obtained by the partial hydrolysis of starch. Partial hydrolysis of starch by an enzyme called diastase. Okay, by the presence of diastase, partial hydrolysis of starch takes place, and maltose is obtained, and it is present in malt. Malt means sprouted barley seeds. Okay, next, structure of maltose. Structure. Its molecular formula by element analysis and molecular weight determination shows that its molecular formula is C12H22O11. Okay, and this on hydrolysis, maltose on hydrolysis, in the presence of an enzyme called maltase, it gives two monosaccharides. Okay, so and these two monosaccharides, that is, maltose is made up of two monosaccharide units, which are two alpha D glucose units. Okay, two alpha D glucose units are present in maltose. Okay, and this contains a glycosidic linkage between C1 of one unit and C4 of other unit. Okay, glycosidic linkages, C1 of one unit and C4 of other glucose unit. Okay. This is the structure of maltose, and this is one monosaccharide alpha D glucose, another one alpha D glucose. These two alpha D glucose units are present, and C1 of one glucose unit form a glycosidic linkage with the C4 of other glucose unit, like this. C5, 6. Okay. Fourth carbon and first carbon are linked together and form a glycosidic linkage. Okay. Next, here in the solution, the second unit gives free aldehyde group. Okay. So it gives the different tests for aldehydes. So this is called as, this is a reducing sugar. Okay. Reducing sugar. Maltose is a reducing sugar. Okay. These different uses of maltose are, it can be used in infant foods. Used in infant foods. And it is seen in malted milk. Okay. Malted milk. Next, third compound is lactose, also called as milk sugar. Okay. And it is occurred in milk of all animals. Milk of all animals. And it is obtained commercially from cow's milk. Okay. Next structure. Molecular determination, molecular weight determination shows that its molecular formula is C12H22O11. Okay, and it consists of two monosaccharide units, and these are obtained by the hydrolysis of this lactose in presence of an enzyme called lactase. Lactase is the enzyme used for the hydrolysis of lactose, and it gives two monosaccharide units which are beta D galactose, and beta D glucose. Okay, and beta D glucose. These two units are linked together, or oxide linkage is obtained, glycosidic linkage is obtained between beta D galactose and beta D glucose. The linkage is between C1 of beta D galactose and C4 of beta D glucose. Okay, and this is a reducing sugar. This is a reducing sugar. Okay. So this is the structure of lactose, and there is a glycosidic linkage between beta D galactose and beta D glucose, and linkages between C1 carbon atom of beta D galactose and C4 carbon atom of beta D glucose. Here we can see that the difference from the beta D glucose is here the fourth carbon, the hydrogen and OH groups are different. This is the difference between beta D galactose and beta D glucose. Okay, so this is the structure, and this can be used in baby foods. Different uses are used in baby foods and in pharmacy. It is used in pharmacy as a base for compressed tablets. Okay, this is the different uses of, these are the different uses of lactose. Okay.